we had to vaccinate almost all the health workers, particularly those along the border with not just Conakry, but with also Liberia. So it's not just about ring vaccination. So we've gone past that now. So now what we are investigating further is to be able to show which of these vaccine should be used as a routine vaccine against Ebola, and if there's an epidemic, which one of them you can readily deploy it to be able to provide immediate response to combat the spread of the disease. So it's not just ring vaccination. So you can use it in real sense.
Thank you so much. I really enjoyed your presentation. It was really informative. I was wondering what was the lessons learned from this project that you could use for combating COVID? I assume that, for example, you mentioned about cold chain infrastructure. It could possibly help you for COVID, but also maybe some regulatory functions that you developed in your country or any other aspects. Thank you.
So there were a lot of lessons learned, not just for the vaccine trial, but in terms of the management of the whole Ebola epidemic, such that when COVID really struck, it was now very easy for the population to understand what we mean by infection prevention and control. And it was also easy for them to appreciate policies that were developed to protect them against the spread of the COVID. Because in the early days of Ebola, when the government developed policies, the community will have to go against those policies. But then with those experiences with Ebola, it was easier with COVID. And also, one of the things that really assisted in terms of combating the spread of Ebola was the recruitment of health community people within their community to help in the fight against the disease. So we actually use people from the community that live with the people, so that once you train them and there is that trust between the people in that community, they will tend to believe them than people coming from outside. So following Ebola, we had to sort of train community health workers. These are they are not former health workers, but we identify people from the community and we give them very simple trainings, particularly in terms of infection prevention and control, and in terms of hygiene and simple things. So these are the people that work within the community and they work with the health workers. So when COVID struck, it was easier for them to just pass on the message, and that actually assisted. And also in terms of regulatory oversight, during the epidemic and when we were about to roll out the vaccine, the regulators were not too comfortable or fair with some of the science of the vaccine. So some meetings were held at regional level or sub-regional level to bring all of the regulators to speed. So there was some amount of training. And because of their involvement also in the vaccine study within the sub-region, it was not just done in Sierra Leone, it was done in Guinea and Liberia. Because of their involvement, they were now like comfortable in terms of dealing with epidemic of that nature, so that when COVID struck, it was easy for those regulatory oversight to be provided. Yeah.
Thanks very much. This is really terrific. And I love the idea of the deferred arm for the controls. which is, in case anybody missed it, was how they did those male circumcision trials for HIV prevention. Could you, I may have missed this, but which antigen was expressed in the VSV, or the VZV, or VSV? And is it strain specific to Zaire, or does it cross the other Ebola strains as well?
It was really strain specific to the Zaire Ebola. So what that meant was that once you have the vaccine, because some of the adverse events will also look like the disease, because there was this approach that once you have a febrile illness, you'll be taken to a old center, and initially that was the source of most of the infection, because it took the lab maybe 3 to 4 days before they actually produced the results. And if you are kept in the old center, then there was a tendency for you to get infected if there were infected cases in the old center. So with the STRIVE, we have to come up with a new definition, case definition of cases that should be eligible to go to the old center. And if you are a candidate for the vaccine, or you're a participant, then if you came down with symptoms that look like Ebola, you will not be taken to the old center. You have to do the test so that it can prevent people from unnecessarily getting Ebola.
Hi, thank you for that talk. I'm Michelle Shang. I work on malaria here. Just a question about just sort of the delays in the trial start. Like, moving forward, are there mechanisms in place where a future trial wouldn't encounter that challenge? Like, maybe they already have a vaccine ready, or maybe even in this new trial you're doing, you have like an IRB already approved for when something's going to happen, to be able to look at effectiveness to prevent infection and not just the immunogenicity outcomes. So that's one question. And then the others are just about malaria. Did you think about just treating people for malaria when they enrolled, or giving some prophylaxis or chemoprevention during their time in the trial, so that you wouldn't see those potential adverse events or obscure the picture? And then on this outcome, looking at if malaria infection is associated with immunogenicity response, how are you measuring malaria? Is it microscopy, PCR, or antibodies? Just thank you.
So let me just answer the last one in terms of the determination of malaria is by PCR, just to be sure. And then in terms of the first question was on the experience. No, well, we really did not give them prophylaxis because malaria is endemic. So if you are going to give prophylaxis, we are going to give all 8,000 or so people prophylaxis. That was also going to increase the cost. Yeah, and most people came in with really, even if there are no symptoms, if you now went to Sierra Leone and you want to check for malaria, most people will have some amount of parasite in their blood, but they will be asymptomatic anyway. Yeah, unless if there is now a stress, or there is something, or they get some more exposure, then they will come down with malaria. So, but we didn't give them prophylaxis. But then in terms of the regulatory oversight and the ethics, some amount of training was done, and also to expedite the approval processes. What we ended up having as an SOP in cases like this, there will be a joint review by the regulatory bodies and the ethics committee, just to sort of expedite the process so that you can deploy the vaccine or whatever within a very short period without losing a large amount of time.
I have a mic. So, thank you. Thank you so much for your really impressive study. I think my question is about the study design, and it's a crossover design with no placebo group, which is very unique, I would say. I took a picture, I want to study more. But two questions: why wasn't the placebo was considered in the study, and how that would affect the results? Because it was at the high course of the epidemic, and epidemics would go away in a few weeks or a month, and then your control group would be in a low epidemic time, and how that would affect the results. So two questions, but again, thanks for impressive study.
Yeah, so
I know FDA and CDC was involved.
yeah,
I know.
I know. The local ethics committee was because at that time all what we had was hope, and one of the things that brought the hope was the vaccine. So the fact that there was a vaccine that is potentially effective to prevent against Ebola, that gave the hope. So ethics was reluctant to approve a study design where people who are enrolled will not get the vaccine. So
But why they didn't allow you to give the placebo? That was my question. Yeah, I know they delayed it, but why did you give the placebo?
Well, I'm not part of the ethics, so I cannot answer that. But they were reluctant to approve. But then they approved this design where, because if you all came from the same community, and then one group, you are living in the same community, one group received the vaccine immediately, and then the other group receive it later. But then the deferred group now is used as a placebo group because you are living in the same community, you have the same exposure risk. So you are not comparing it to another community. You are comparing it to the community where they are coming from. So you have the same amount of exposure. So that was what you use as a placebo. But then the efficacy was really designed to be estimated within the immediate group. Because once you cross over, then there is no other group to compare you with. But then in terms of adverse events, you were being followed up, and then adverse events were documented.
Hi, I was wondering, you said in the beginning you excluded those that had an HIV positive diagnosis, but towards the end that there are further studies assessing the safety and efficacy for those that have malaria or a helminth diagnosis. Are there any studies looking into the efficacy or safety for HIV positive patients, especially those with an adequate CD4 count or even that have undetectable viral loads?
But not at the moment in Sierra Leone in terms of HIV. But then at that time, because we really did not have enough data, so one was not going to take the risk because everybody was interested really to come up for the vaccine. So one was not going to take the risk to give it to HIV patients because we did not have enough information to support the use of the vaccine in HIV patients.
Thanks. Thanks for the talk, Mike Chirange. I could have missed it, but I wanted to know if there was any, for pregnant cohorts, what did you do for pregnant women in terms of your IRB approval during your study? That's the first part. And the second part is like in vaccine development for HIV, there's this unique epitope antigen reaction we see called vaccine-induced seropositivity. In other words, these are people you offer HIV vaccine, and the antigen of that vaccine will create an antibody that when you are testing the HIV, even if they are not infected, they will test positive. So did you experience that? I have no knowledge in terms of the antigen and epitope for Ebola. Do you experience that? And the first part was the pregnancy cohorts. And how acceptable was the vaccine if you indeed enrolled pregnant women?
Well, pregnancy was one of the exclusion criteria anyway, and but that was rarely reported if you are not visibly pregnant. But then we ended up having people who became pregnant during the follow-up period, and they were followed up anyway throughout the pregnancy, and there was no significant outcome in terms of effect on the mother and or the baby. But in terms of your question on HIV, I really cannot answer.
It was more like on your Ebola testing. I think you made reference to during the study itself, somebody who received the vaccine could go to a treatment center with fever and joint-like illness, and you do a test. That's the point. You do a test.
Yeah.
And sometimes cross-reactivity, if you don't have a good assay, cross-reactivity can result in a positive test where there's no disease. That's what I meant.
In terms of false negative.
Correct.
Yeah. Well, particularly for those that were kept in the holding center, if they were false negative, but then they were in the holding center, then they became the source of the infection for others. So that was a bit of a problem.
Dr. Samai, thanks so much. I had just a question to follow up on the pregnancy. There was one patient who died in pregnancy under one of the deaths, and maybe wasn't pregnant at the time that they enrolled in the trial. Do you know, did they follow to see exactly what caused her death during pregnancy?
Well, I cannot tell you now from the top of my head what was the cause of the death, but what I know is that it was not really vaccine related. But I cannot tell you the exact cause of the death.
A few minutes over, but I just want to thank you, Dr. Samai. Fascinating, important work, and thank you for all the questions. It's a real privilege to have you here at UCSF. All right, thanks everyone.